My name is Hannah Rigoni, and I'm a recent undergraduate from New Mexico Tech. I've been working as the NAGT student this summer at the U.S. Geological Survey in Boulder, Colorado. Today, I'm going to be talking about the impacts of wildfires on microbial communities in the Russian River watershed in Northern California. Wildfires burned in Northern California in October 2017. Sonoma County Water Agency lies within the Russian River watershed, which supplies water to over 600,000 people. The Russian River watershed is partially located in these burned areas. Consequently, there is concern for water quality and contaminant release in the impacted areas. Changes could alter microbial communities and biogeochemical cycles. This brings about concerns for the bank filtration site, such as harmful algal blooms, clogging, or other water quality issues. Bank filtration is a primary form of water treatment where surface water is drawn down through the aquifer sediments and into collector wells. To better understand the relationship between biogeography, water quality, and wildfires, this study looked at temporal and spatial variations in microbial communities against water chemistry, drainage area size, and other regional differences. A dedicated field crew from Sonoma Water and Lawrence Berkeley National Lab graciously braved conditions immediately following rain events to collect water from 11 different sites during the post-fire rainy season, which extends from October 2017 to April 2018, and from the dry season, which in this study, we have analyses up to June 2018. We analyzed DNA sequences, DNA yield, and nutrients such as carbon and nitrogen, and field parameters such as turbidity and trace metals. However, here we only present constituents that most closely are correlated to variations in microbial communities. Here's a map of the Russian River watershed, which includes our 11 sites, and the fire parameters from October 2017. The Wohler site is located above an inflatable dam that's deployed during the dry season. It's sampled only during the dry season. The Mirabel site is located directly below the inflatable dam and is sampled year-round. Other sites along the Russian River include Jimtown and Cloverdale. The sites associated with the Tubbs and Nuns fire are Makama Creek, Franz Creek, Mark West Creek, Brush Creek, and Matanzas Creek. The pocket fire is associated with the Big Sulphur Creek. The Russian River, Capella, and West Fork sites are closely related to the Redwood Valley fire incident. You might notice that all the fires are located along the eastern side of the Russian River watershed, and that's because it's drier. This side, the western side, has mountains that get higher precipitation and are encompassed by redwoods, which don't burn easily. This first figure illustrates the differences in alpha diversity using the Shannon Diversity Index, which is a quantitative measurement of community richness. The x-axis depicts the sites and the y-axis depicts the diversity index, with higher numbers showing more diversity. The colors show associated wildfire areas. We found that samples from the different fires had no significant differences in diversity from each other, or the unaffected tributary. The Wohler site was found to have the lowest diversity, and the site directly below it, the Mirabel, also had low diversity, but it was more variable due to the fact that there are sampling events from when the dam was down during the rainy season. This figure illustrates beta diversity using a principal coordinate analysis plot based on weighted unifrac dissimilarities, which is a measure of dissimilarity that incorporates phylogenetic relatedness and abundance. The color of each circle corresponds to associated wildfire area, and the size corresponds to the drainage area in square miles. The green points clustered over here depict the samples related to the main stem of the watershed and have large drainage areas. These small points in shades of red and orange and blue are in the smaller tributaries. Within the smaller tributaries, there is a separation by fire. The Tubbs and Nuns fire are generally cluster higher on the y-axis, while the redwood and pocket fire cluster lower. And the control tributary seems to cluster in the middle of these. You'll also notice that the points associated with the mid-main stem cluster in the generally closer to the tributaries. Interestingly, there is no large temporal variations observed thus far in our sampling program. In order to better understand which water constituents had the largest relationship with microbial community compositions, a conical correspondence analysis was performed. The green circles on this plot correspond to different sites. Squares are abundant taxa, the red triangles correspond to associated fire areas, and finally the blue vectors are the factors that have the strongest relationship with variability in microbial community composition. From this, we can see that the main stem below the dam has a large correlation with the size of the drainage area, but negatively correlates with the other constituents. Sample sites that resided within the redwood and pocket fires have higher turbidity and higher concentrations of aluminum and iron, indicating higher amounts of sediment load from these drainage areas. The sites that were affected by the Tubbs and Nuns fire have higher biomass represented by DNA yield and higher DOC, phosphorus, and manganese constituents. In conclusion, microbial community structure is related primarily to the size of drainage area and secondarily related to regional water quality differences. Contaminants released by wildfires can vary on regional differences in land use and vegetation. The sites with higher transported biomass also had higher phosphorus, DOC, and manganese. The inflatable dam lowered diversity. We are now ending our first year of sampling and will include sites impacted by the more recent Mendocino wildfire. And we will continue to ascertain whether there are differences in microbial communities and functions resulting from the fires. Special thanks to Sonoma Water for funding, Dave Roth Lab for trace metal analyses, and Michigan State University for Illumina sequencing. And special thanks for listening.